human genetics pursuing these types of studies. In this webcast, we'll first hear from Gosia Trinka from the Wellcome Sanger Institute, who will discuss sequencing approaches for prioritizing GWAS variants associated with immunological diseases. We will then hear from Nadav Ahitov from UCSF, who will discuss strategies for identification of causal genes and exploration of disease mechanisms using genetics, sequencing, and massively parallel reporting assay techniques. After these short presentations, stay tuned for a live Q&A moderated by editors Tiago Fael and Catherine Matensky from Nature Genetics, where the experts will answer questions from the audience. If you're listening to this webcast live, just type your question in the text box on the screen at any time during the discussion, and we'll try to answer as many questions as possible. And with that, I will turn it now over to Gosia Trinka.
Hello, my name is Gosia Trinka. I run the Immune Genomics Research Group at the Wellcome Sanger Institute, where we study how genetic variants regulate the immune system and predispose to immune diseases. I'm also the Director of Experimental Sciences of Open Targets, which is a public-private partnership where we use human genetics and genomics to systematically identify drug targets. Today, I'll share with you some of our recent work on translating immune disease GWAS variants to function. Common complex immune diseases collectively affect about 10% of the population and can manifest through different ways and affect different organs. However, they have one feature in common: the overreactive immune system. From the genetics perspective, immune diseases have been at the forefront of genome-wide association studies, and as a result, there are currently thousands of identified disease risk variants. Yet how these variants lead to impaired immune system is not very well understood. All the efforts to translate GWAS variants to function must start in disease-relevant cell types to capture the variant effects in the right context. In the case of immune diseases, this is somewhat challenging as there is a broad spectrum of immune cell types. And these immune cells can actively, in fact, they do actively communicate with each other, and they dynamically transition through different cell states, for example, in response to inflammatory cytokines. Yet once the disease-relevant cell type is identified, we can then focus on efforts on scrutinizing the potential molecular mechanisms of disease-associated variants in this specific cell type to identify disease-causal genes, disease-causal variants, and more broadly, the regulated pathways and cell functions. One of the approaches that can leverage the GWAS information to help nominate disease-causal cell types is the integration of GWAS variants with chromatin marks. In such approaches, one can measure if disease-associated variants overlap with chromatin-accessible regions or histone marks that highlight active promoters and enhancers. If the variants are residing within chromatin mark peaks and the observed overlap is significant, then this could pinpoint a disease-relevant cell type. Few years back, we developed a statistical framework for SNP enrichment analysis, and we applied it across different cell types and tissues. We observed that variants associated with immune diseases were highly enriched among specific immune cell subsets, especially across T cells. And in particular, one cell type, the regulatory T cells, was enriched among many immune diseases. The general role of T cells is to direct the immune response. In the presence of an antigen, T cells are activated and start proliferating to drive the inflammation. Tregs are a subset of CD4 T cells. They are infrequent, only about 5% of CD4 T cells, but they play a critical role in regulating the immune system because they suppress the immune response. They can do it directly by interacting with other immune cells or indirectly through secreting cytokines. However, these low numbers of Tregs in the circulating blood make them challenging to study as they are hard to collect in sufficiently high numbers for large-scale genomic assays. As we were convinced that the genetics was pointing us to a relevant cell type, we set out an ambitious project to profile Tregs across over 100 individuals so that we were able to perform QTL analysis. QTL mapping is most frequently applied to transcriptomics data to map the effects of variants on gene expression. But equally, it can be applied to chromatin marks to, for example, identify allelic effects on chromatin accessibility or enhancer activity. Together with gene expression, it allows to map the allelic effects on regulatory elements and on the target genes. Additionally, by identifying variants that directly overlap with chromatin QTLs, the approach allows to suggest plausible functional variants. And finally, through colocalization of QTLs and GWAS signals, we can define shared signals that could be driven by the same causal variants. We used Tregs isolated from about 120 individuals and generated QTL maps of chromatin activity and gene expression. We used 3 different chromatin marks and observed the highest proportion of QTLs in H3K27 acetylation, a mark that highlights active promoters and enhancers. Interestingly, for nearly 80% of eQTLs, we were able to link E gene, an eQTL gene, to a chromatin feature. On the other hand, we were unable to link the majority of acetylation QTLs to eQTL effects. We observed 133 unique GWAS loci that colocalize with immune diseases and different QTLs in our Treg dataset. For only a quarter, we identified colocalizations to both eQTLs and chromatin QTLs. When we compared these results to QTLs from closely related naive T cells, we observed that some of our colocalization events were shared between the 2 related cell types, and some were specifically pointing towards dysregulated Treg function. Now, on the right-hand side, what you can see is that by overlapping GWAS signals with our chromatin QTLs, we were able to substantially refine the numbers of disease variants to smaller sets of plausible functional variants. This dataset generated a rich resource for us to derive hypotheses and study the role of candidate variants and genes in more detailed functional follow-up experiments. Such high-throughput approaches are essential to functionally refine the GWAS associations down to numbers of functional variants and genes that are feasible in more extensive functional follow-up studies. One of the unexpected results from this study was the observation that nearly 40% of colocalizations were detected only with chromatin QTLs without a link to an eQTL. On the right-hand side is an example of Treg-specific enhancer QTL that colocalized with a number of immune diseases, including inflammatory bowel disease, type 1 diabetes, asthma, and psoriasis. We teamed up with Rahul Raichowdhury's group at the Babraham Institute near Cambridge and followed up this colocalization in greater detail. Rahul's group has located and deleted a corresponding enhancer region in mice. They observed that this deletion affected the expression of a nearby gene, LRRC32, encoding the GARP protein. In knockout mice, the expression of GARP was specifically affected in Tregs upon cell activation and not in a resting state. More extensive phenotyping showed that mice with the enhancer deletion were most susceptible to developing colitis, and the Tregs had impaired suppressive capacity and were unable to appropriately control inflammation. Finally, when we investigated 3D structure in this region, we observed that there was indeed a Treg-specific contact between the LRRC32 enhancer and the gene promoter. Using activating the Cas9 experiments and guides targeting the fine-map variants residing within the enhancer, Daria's group was able to define a small regulatory region that contained one of the LD variants and showed that this region controlled the expression of GARP. This extensive follow-up study to the colocalization of a Treg enhancer QTL reinforces the concept that at least a proportion of GWAS variants will function through a cell type and cell context specific mechanism and may require tailored functional approaches to identify such effects. For immune cells, the disease-relevant context could manifest at different stages of cell activation or in response to the presence of specific cytokines in the environment. Upon activation, T cells polarize towards different subsets of T helper cells and exert different functions, for example, through interactions with other immune cells. This polarization process is driven by the presence of different cytokines in the environment. In another project, we set out to comprehensively identify immune cell states most relevant to immune diseases. We used naive and memory CD4 T cells and macrophages and exposed the cells to cytokines that are linked to autoimmune diseases and inflammatory processes. We then profiled chromatin activity at 2 different time points, capturing early and late cell activation events. Collectively, we generated data for 55 different cell states. We observed that stimulation in general induced profound changes to the chromatin, which you can see on the right-hand side of the plot. However, for the majority of the induced peaks, we observed that they were shared across the cytokine conditions. Now, despite this sharing, which you can see on the right-hand side of this plot, we observed that the chromatin peaks expressed high variability in their activity across the different cell states. This had implications to our ability to test for SNP enrichment. Typically, the SNP enrichment methods measure the overlap between variants and peaks within a cell type. When the overlap between peaks across cell types is relatively low, this allows to identify enrichment in peaks present in specific cell types. In the case of our dataset, we were dealing with closely related cell types, and the chromatin changes induced by the cytokine stimulations modulated the activity of the peaks rather than their presence or absence. So to address this, we developed a new SNP enrichment method called CHEERS that takes into account the variable chromatin activity levels. When we applied this method to our dataset, we observed that across different immune diseases, there were individual cytokine-induced cell states that were significantly enriched with the associated variants. However, more globally, we saw that it was predominantly the signal in early activation of memory T cells that capture most of the signal, which is depicted here in the lighter shade purple. This observation was also recapitulated when we ran pathway analysis using genes in proximity to the peaks driving this enrichment. So overall, these results point towards the dysregulation of processes underlying early activation of specifically memory T cells by the variants associated to different immune diseases. And I want to close my talk by sharing some thoughts on the next challenges in following up these results. While we often tend to refer to a cell type based on expression of a handful of cell markers, this is a simplified representation. More often, we deal with cells transitioning from one cell type or state into another. And this is becoming especially apparent as we investigate single cells at the transcriptional level. This continuum of cell states is something we will need to start considering in our variant-to-function efforts. Here, I show one of our recent projects where we used single-cell RNA sequencing to investigate gene expression regulation of naive and memory CD4 T cells. We observed that transcriptionally, the cells formed a continuum which was driven by the increased expression of effector genes such as cytokines and chemokines. So, rather than defining specific cell types, we assigned cell states based on the position of a cell on this effectiveness gradient. When we measured the response of cells to external cytokine stimuli, we observed that for some genes the expression was dependent based on the cell effectiveness status, and in some cases we observed an interaction between the effectiveness and cytokine stimulation, such as, for example, for IL-9, where gene expression was increasing with a cell's effectiveness only in Th17 and iTreg polarizing conditions, and not upon activation in the absence of any cytokines or in the presence of Th2 polarizing cytokines. So I'll close it here. These are the superheroes behind the work in the group. I want to especially acknowledge Daphne and Lara, who led the enormous efforts to generate and analyze the Treg data, and Blagoje and Eddie for scrutinizing processes underlying T-cell activation. I'd also like to thank my collaborators who bring in invaluable immunology expertise to these projects and are brave to take on extensive functional follow-up studies with us. And finally, I'd like to thank the Sanger and Open Targets for funding and supporting our research. Thank you to Catherine and Tiago for inviting me to take part in this webcast, and I look forward to the discussion.
That was Gosha Trinka, and I will now hand it over to Nadav Ahitub.
So my lab is interested in gene regulatory elements, in particular in enhancers. Here, for example, you could see a mouse embryo on the top right, and where you have dark blue is where it's expressed, in this case in the limb, in the neural tube, and in the brain. And what people have seen is that the promoter itself is not enough for this expression pattern. What you need are DNA sequences that could be very far away from the gene, called enhancers, that tell it to turn on in the limb, in the neural tube, and the brain. And so the enhancers could be dubbed as the promoters of the promoter. Nucleotide changes in these enhancers and other regulatory elements can lead to differences in gene expression, and those could lead to phenotypes like human variation, human disease, or morphological differences between species, and we as a lab are interested in all of these. And for today's talk, I'll mainly focus on human disease as that is most relevant. So we're really at revolutionary times where the number of genomes that could be sequenced is going up quite substantially, and the sequencing costs are going down quite significantly. However, whenever we sequence a human genome, only 2% of it codes for protein and is genes, while 98% is non-coding. So we have this vast genomic space to search for regulatory elements. So in that top right box, if that was the human genome, only these two red base pairs actually code for protein. And in terms of variation, whenever we sequence a human genome, we get on average 3.5 million variants. Amongst those, only 0.65%, that small little red line on the C there, are the variations that we find in coding regions, and everything else is non-coding. For genes, we have a nice code that we know. For example, CCC codes for proline, and we know the consequences of that change. If we change CCC to CTC, proline will change to a leucine. But we don't have all that in regulatory elements, and even if I was to find a mutation in a known enhancer, for example, I really currently don't know how to tell phenotype from genotype. And that is most illustrated from the most characterized enhancer to date, and that is the sonic hedgehog limb enhancer that leads to mutations in humans and in mice that cause limb abnormalities. So sonic hedgehog, the gene, as you can see here on the top left, and mutations in the gene itself lead to holoprosencephaly, cyclopia, kids are born with one eye, limb defects, a lot of bad things. And then one megabase away from sonic hedgehog in an intron of another gene is the sonic hedgehog limb enhancer. And if you remove it in mice, you can see on the bottom right that mice where this enhancer were removed have a limb truncation phenotype versus the mice on the left, which are the wild type littermates. If you sequence it in patients who have an extra finger, polydactyly, you can see here two different cases of polydactyly that segregate perfectly with the family, suggesting that these point mutations lead to limb malformations in these families. And now there's over about 20 different point mutations in this enhancer that lead to limb malformations. So we could now ask the question, can we tell phenotype from genotype for these mutations? And the short answer, sadly, is no. And just to illustrate that, I'll show you an example from a big Swedish family that came into our lab. They had an extra finger, and so the first thing we do is sequence it for the sonic hedgehog limb enhancer, and to our surprise we find a 13 base pair insertion in this enhancer that leads to exactly the same phenotype as a point mutation. Now that was quite surprising for me, as I would expect an insertion to be a much more severe phenotype, but yet you get exactly the same mutation. So the next step is to try to characterize what this mutation might be doing. And so we could look at transcription factor binding sites, and we could see that a lot of them are inserted, and not any are deleted, but that's all circumstantial. We really don't know how that leads to this phenotype. We could do a mouse enhancer assay where we take a sequence that we think is an enhancer and we throw it in front of a minimal promoter. This is a promoter that's always off, like a light switch that's always off. If it has an enhancer in front of it, it will turn on that light switch and turn on the reporter gene. And we can stick this into transgenic mice and compare it to exactly the same sequence, but now with the 13 base pair insertion, and ask the question, is there a difference between them? And when we do that, we see the wild type here on the top and the 13 base pair insertion below it, and you can clearly see that there is a ectopic expression of this enhancer that likely may be leading to that extra finger. So that's great, but there's a lot of things that are problematic with this assay. One, it's not quantitative. Each embryo gets a different number of integrations of your transgene, so you can't measure it. And in addition, when we look, for example, at two different point mutations that lead to a much more severe phenotype, where it's not just an extra finger, but also the radius and the ulna that are affected, the legs that are affected, and we model them using this assay, you can see, again, ectopic expression, but it really doesn't tell me why this mutation is more severe in terms of a phenotype versus this mutation here. So what we're stuck with now is tons of genomic data, a limited understanding of the regulatory code, and then we have this limited assay that's not really quantitative, and in addition, not high throughput, so I can't really measure all the thousands and thousands of variations that we're getting in the genome and the thousands of regulatory elements that are there. So about 10 years ago, we teamed up with Jay Shandour's lab to really develop this assay and make it more high throughput. And the work that I'll show you initially is alongside with Greg Cooper at Hudson Alpha and Daniela Witten, who's in UW, spearheaded by Fumitaka Inawa, a very talented postdoc in my lab, and Martin Kircher, a very talented postdoc in Jay's lab, who now has his own lab in Berlin. So going back to this assay, where you have a sequence in front of minimal and reporter gene, we really wanted to make it more high throughput, and the idea was to stick in a 15 base pair barcode that makes RNA. And so if this sequence is an enhancer, it will turn on the minimal promoter, turn on the reporter gene, but will also provide you with a 15 base pair barcode that could be read via next generation sequencing technologies. So now what we could do is clone thousands of sequences on the left and thousands of sequences on the right, and with paired end reads, where you sequence the same DNA sequence from the right and from the left, you can assign the barcode to the enhancer. So we can take a library of hundreds of thousands of sequences and throw it into cells or into mice, measure DNA to see if the barcode went into the cell or the mouse, and measure RNA to see if it turned on the minimal promoter and reporter gene, and we can plot that over RNA over DNA to measure the effect. So for example, this red sequence here in the table, we see both DNA, meaning it went into the cell and into the mouse, and we also see a lot of RNA, suggesting that it's a strong enhancer. For the yellow one, we only see DNA, meaning it went in, but we don't see RNA, meaning it did not turn on the minimal promoter and reporter gene, and likely is not an enhancer. And we've published over the years quite a lot using this technology, is how we can take this technique and use it to better understand the regulatory code. The idea being, if there's a language that we don't know, we can either take known words that we know and mutate them to death and see what works, what doesn't, or the opposite, build a language from scratch and see what works, what doesn't, and get a better understanding of grammar. And so I'll start with the saturation mutagenesis, whereby we can take a known promoter or known enhancer, for example, mutate it to def. For example, if it has an A in a certain position, change it to a C, to a T, or a G, and then see what kills a function or makes it work better due to these mutations. So for this project, we chose 10 promoters and 10 enhancers. You can see the names of the genes on the left that lead to various diseases, where you can see the names of the diseases on the right. And basically generated libraries of over 30,000 different mutations to see how they affect promoter or enhancer activity. As an example, I'll show you the telomerase promoter that was associated with various types of cancer. Mutations in either position -146 or -124 in the telomerase promoter have been associated with specific types of cancer. For example, in glioblastoma, 83% of the patients will have either mutation -146 or -124. 71% of melanoma cases will have either one of these. And these are activating mutations. They make telomerase active, leading to cancer progression. So we took the telomerase promoter, did all possible mutations, and what you can see here, 0 is the line of the wild type activity, and you can see below it mutations that reduce promoter activity and mutations that increase promoter activity. And if you look at where -146 or -124 are, those are exactly the red arrows here that lead to increased promoter activity. In addition, you see a lot of mutations that are sometimes also found in patients that are not -146 or -124. And so by having all of these mutations, this is extremely helpful for clinicians who find novel mutations. And in addition, you see mutations that reduce activity. And if we could figure out, for example, what transcription factors might be binding there, we might be able to use antagonists for them as treatment for this patient. And finally, having 30,000 different mutations can really allow us to predict and generate better biological tools that can predict how mutations in gene regulatory elements lead to disease. So the opposite approach could be to build a language from scratch and see what happens. And so here we took neutral template sequences that we knew are not enhancers and start spraying transcription factor binding sites on them to see how they lead to enhancer activity. And so here we chose 12 different transcription factor binding sites that you can see in the table on the left. And using them, we modeled 5,000 different 168 base pair sequences in various ways. We took 2 neutral templates, 2 different sequences that we know are not enhancers, and then we either put 1 transcription factor binding site on them, either on the right or on the left, as you can see in the red boxes there. We could put 2 transcription factor binding sites and play with distance or the combinations or number of them. And finally, as you can see all the way to the right, we can take, for example, 3 transcription factor binding sites and just play with order to see how order can affect their enhancer activity in this case. So here we actually used a technique called the hydrodynamic tail vein assay, which basically what we do here is inject a large volume of liquid in a very short period of time, 4 to 8 seconds, into the mouse tail vein. And that, after 8 to 24 hours, leads to a very strong specific expression in the liver. So it's a great way to get DNA into the liver. It actually doesn't work well for other tissues. And so our first observation here was the more transcription factor binding sites you have, the stronger you are, as you can see here. In terms of 1 transcription factor, we can see, as you can see for the top, that for some transcription factors, if we increase the numbers of transcription factor binding sites on the sequence, we get stronger activity. While for some, if you look at the ones below, we don't see an effect of more transcription factors on strong expression of the enhancer. For 2 transcription factors, what was nice is we could look at interactions between different transcription factor binding sites, and where you see red lines, that means that there is strong interactions between them, suggesting that if you have both transcription factor binding sites together, you have a much stronger enhancer activity than having each one separately. While if you have a black line, that means that they actually interfere with one another and reduce enhancer activity in this case. We wanted to see if what we observed in terms of grammar actually exists in real life. Everything we've done here was synthetic. And so we looked at ChIP-seq data sets for the liver that characterize 52,000 potential enhancers in the liver. And when we look to see if those interactions that we see on the bottom left exist much more than what you'd expect by chance in these real-life enhancers, we see that at least 7 out of the 8 of them do significantly exist much more than what you'd expect by chance in these real-life enhancers, suggesting that what we see is actually real in actual enhancers. In terms of order, as I mentioned, we've done quite a lot of testing on order where we shuffle around sequences. Here I'm showing you 8 different sets of transcription factors that are shuffled 49 different times. So basically we take 8 different transcription factors and we shuffle them in 49 different combinations. And what's interesting here is that if we compare the sequence on the top versus the sequence on the bottom, we see a 6-fold increase in expression for the sequence on the top versus the one on the bottom, suggesting that order is important for activity here. So just to sum up this part, not all transcription factors increase activity. If you have heterotypic sequences, or sequences that have different transcription factors, you tend to be stronger. Certain transcription factors work with one another and some interfere, and order has an important effect on enhancer activity. And finally, everything that we've observed, we see also in real-life enhancers, suggesting that these grammatical rules exist in the wild. Finally, I just want to end with this slide where we've actually modified this assay a few years ago to work with lentivirus. And the advantage there is that we can infect hard-to-transfect cells like neurons or others. And in addition, we also have an integrated readout where lentivirus integrates into the genome. And so we have an in-genome readout of our MPRA, which we feel is a bit superior to episomal. And all of our work now that we are doing in the lab is using this lentiviral MPRA approach. With that, I want to end with the most important slide, thanking members of my lab and our collaborators, in particular Jay Shandur from the University of Washington and Martin Kircher from the Berlin Institute of Health, and our funding sources. Thank you very much for listening.
That was Nadav Akituf. Thank you so much for the great presentation, Gosha and Nadav. My name is Catherine Potensky, Nature Genetics, and we'll now start the live question and answer portion of this webcast. As a reminder, please feel free to submit your questions if possible. So we'll just right now. So to start, a variety over an array of systems, and Gosha, we touched on this a little bit about the future, but I wanted to ask, what technologies are you excited about for solving these problems of getting at regulatory variation and how we're going from that sequence to phenotype? Gosha, we can start with you and then...
Yeah, hi, thanks, Catherine. I'm actually really excited about but I think what I was trying to show is that really so important. So I guess the technologies I am mostly excited about are the ones that will allow us to test different in primary cells in the
Definitely. Nadav, do you want to comment on what technologies you're excited about?
Yeah, I completely agree with Gosha that we really have to get this into a single cell manner because there's very tissue specific do this in bulk. And so, really excited about getting this to work in